Okay. Dear colleagues, let's start the procedure. Today's defense is transcriptomic analysis of the interaction between pre-mRNA splicing and intronic polyadenylation by Mariia Vlasenok. And let me start with presenting the jury members. Dr. Jonathan Jin, full professor at the College of Life Sciences in Zhejiang University and an adjunct professor at the Shanghai Institute for Advanced Study of the same university in China. Dr. Jonathan Jin graduated from Zhejiang Agricultural University 6 years ago and became an associate professor in the College of Life Sciences there. And then he was promoted to full professor several years ago. He has a research group that whose interests are in RNA processing, editing, functions of non-coding RNA structure, and currently he works on long-range RNA interactions and their role in alternative splicing. And it's very close to the interests of Professor Pervushin, so I assume that they know each other well. He has published about 100 research papers. Our next external member is Dr. Oleg Gusev, the head of Department of Regulatory Transcriptomics for Medical Genetic Diagnostics and Jintendo University Graduate School of Medicine in Tokyo. He also has a lab at Kazan Federal University. He got his PhD from Okayama University in 2007. And his research interests are mainly in biology of extremophiles, I think. Other things are listed in the presentation, but I know Professor Gusev for several years now because of the paper, and his real life, of course, is biology of extremophiles. And again, he's publishing very well, and he's doing extremely interesting science. Our next external member is Dr. Ivan Kulakovsky, a leading research scientist and professor at the Institute of Protein Research in Pushchino, and a senior researcher at the Engelhardt Institute of Molecular Biology, and also at Vavilov Institute of General Genetics, and again in Kazan Federal University. He's got his PhD in bioinformatics at our university. Actually, I was the chair of dissertation council under supervision of Professor Tumanyan, and then he got his Doctor of Science degree again at IITP. And his main research interests are regulatory motifs, binding sites of transcription factors and their role in regulation of expression. And he has developed a number of very important and useful tools and databases in this area. And now we turn to our local member, Professor Ivankov here, an assistant professor at the Center of Life Sciences. He graduated from Moscow Institute of Physics and Technology and studied protein folding under Professor Finkelstein at the Institute of Protein Research in Pushchino. Back 10 years ago, he studied signal peptides at the Technical University of Munich with Professor Frischmann. And then he worked with Fyodor Kondrashov in Barcelona on analysis of epistatic interactions. His main research is in protein folding, specifically in kinetics of protein folding and thermodynamics of proteins. He got Academia Europaea Prize for Young Scientists in 2009. Professor Khraneva, who is stuck in a traffic jam, but hopefully will join us. She's she has joined us online. She graduated from Moscow State University under Professor Perushin and myself. And then she worked for IITP, where she defended her PhD thesis on chromatin structure and alternative splicing. And he worked as a bioinformatician in University of Julius Liebig University of Gießen, Helmholtz Zentrum in Munich, Leiden University Medical Center, and she spent her postdoc with Professor, not spent, actually, she worked as a postdoc with Professor Haitovich in the Partner University for Computational Biology in Shanghai, and then returned to Russia and became a researcher and then an assistant professor here at Caltech. And her main research interests, well, she has actually extremely diverse research interests ranging from transcriptomics, alternative splicing, to lipidomics, to the analysis of chromatin three-dimensional structure. And presently she's working on integrating all these approaches to study how genes work in the human brain. She has won several competitions. Specifically, she was awarded a L'Oreal Prize for Young Women in Science several years ago. She has published a number of very important papers, several papers with Nobel laureate Svante Pääbo, by the way. And that's it. Myself, I'm director for Center of Life Sciences and vice president for biomedical research here at Caltech. I'm not a professor at Moscow State University, as stated here. And I'm not vice director for science at the Kharkiv Institute for Information Transmission Problems, as stated here. But indeed, I am the head of research training and bioinformatics there. My main research interests are in molecular evolution, comparative genomics, many areas related to that. Well, and I have a decent publication record, yes. Our candidate, Mariia Vlasenok, is a PhD student at Professor Perushin's lab here in Caltech. She got her bachelor degree at the Moscow Institute of Physics and Technology. Her initial research was on physics and chemistry of non-canonical DNA structures. And then she joined Caltech Master School in Life Science, graduated there with honors, and then joined the PhD school here. And she's now studying what? I think it was something else. Okay. Anyhow, her PhD research was on splicing and polyadenylation. We'll hear that. Professor Perushin, associate professor, the Center for Molecular and Cellular Biology. She has actually master's degree in mathematics, master degree in chemistry, and PhD in mathematics in algebra. She also has a master degree in bioinformatics in Boston University. And his postdoctoral research was on computational neuroscience. After that, he Moscow and studied developed algorithms for prediction of RNA secondary structure applied these algorithms to study long-range interactions that influence alternative splicing. He worked with Dr. Guiga at Center for Genomic Regulation in Barcelona, working on big data transcriptomics, and he has published several Nature papers as a first author in collaboration with ENCODE Consortium. And the research in his lab here at Caltech is mainly dedicated to various aspects of splicing. And that said, we can now start the presentation. So Mariia, please.
Thank you. No? Hello, dear colleagues. Today, I want to present you my PhD project called the Transcriptomic Analysis of the Interaction between pre-mRNA splicing and intronic polyadenylation. What?
Even if you didn't want, you would still have to.
Yes. Okay, so I'll start with some background after I make the pointer work. Okay, so I have this point there. The majority of mRNAs generated by RNA polymerase II in eukaryotic cells are cleaved and polyadenylated at specific sites called the polyadenylation sites. These polyadenylation sites are defined by RNA sequence elements surrounding the sites. The most prominent of these sequence elements is polyadenylation signal. It's an AAT-AAA hexamer, which is upstream of the polyadenylation site itself. More than 70% of human genes have multiple polyadenylation sites and thus undergo alternative polyadenylation. The additional polyadenylation sites can be located in the same 3' UTR region. Then the alternative polyadenylation is referred to as 3' UTR alternative polyadenylation. And the isoforms generated by this type of alternative polyadenylation can differ in their subcellular localization, mRNA stability, and interaction with microRNAs and RNA binding proteins. Additionally, the additional polyadenylation sites can be located upstream of the terminal exon in the intronic regions. Cleavage and polyadenylation on these intronic sites result in proteins with different C termini, and thus intronic polyadenylation can substantially affect the function of the protein due to its domain loss. About 20% of human genes undergo intronic polyadenylation. However, many more have cryptic intronic polyadenylation sites. Interestingly, research shows that cleavage and polyadenylation machinery recognizes these cryptic intronic polyadenylation sites. However, as we know, they are not used. So this raises an interesting question about the mechanisms of silencing of these intronic sites. Another crucial process of pre-mRNA processing is splicing. As well as cleavage and polyadenylation, it often happens co-transcriptionally, and thus these two processes can interact with each other. There are several evidence for this interaction. First, many known splicing factors serve both during cleavage and polyadenylation and splicing. These include U2 auxiliary factor, PDPP1, and members of HU protein family. Additionally, cleavage and polyadenylation and splicing can alter each other's efficiencies. Specifically, these two processes enhance each other's efficiency during the definition of the terminal exon. However, they compete within individual introns. Specifically, any intronic polyadenylation site can either be a position of cleavage and polyadenylation and thus become an end of truncated transcript. On the other hand, if the intron is spliced out, then this intronic site is degraded as part of the intron layer. There are several methods for polyadenylation sites identification. The most common The most specific and sensitive one are specific protocols capturing 3' ends of transcripts. These protocols use oligo-DT primers. However, since these datasets are not universal and cannot be used for other purposes, there are not many 3' end sequencing datasets. Thus, there's been attempts to identify polyadenylation sites from much more common data type, specifically RNA sequencing. Most computational methods to identify polyadenylation sites from RNA sequencing data use read coverage and look for abrupt decrease in read coverage to identify the polyadenylation sites. However, the read coverage along the gene length is normally non-uniform, and thus these methods work only in the untranslated regions where the original polyadenylation sites, which is distal polyadenylation site, is located. This means that these methods are not very useful for intronic polyadenylation sites identification. On the other hand, RNA sequencing data also contains reads that come from junction between the end, the end of the terminal exon and the poly A tail. These reads have a run of non-templated adenines, which allow to identify the polyadenylation sites. However, the main problem of this approach is that there are less than 1% of these reads in the RNA sequencing datasets. Thus, previous attempts to use poly-A read-based methods to identify polyadenylation sites were not successful since they were applied to relatively small datasets. However, recent appearance of large RNA sequencing datasets created by consortia may be a new step for poly-A read-based approaches. Specifically because even 0.1% of huge amount of reads is a good amount of reads to identify, to base an analysis on. In this specific project, I work with genome tissue expression dataset, which was created by GTEx consortia and contains more than 10,000 RNA sequencing samples from healthy human donors. It covers more than 30 human tissues, and which is most important, has more than 500 billion reads, meaning that potentially there should be about 500 million poly-A reads, which is a good amount to base the analysis on. Additionally, RNA sequencing datasets are good in terms of that they are universal, and thus we can analyze other pre-mRNA processing steps with them. For example, and which would be useful for this research is that it is possible to analyze alternative splicing based on this same dataset. Accordingly, the goal of my project was to systematically examine the interplay between pre-mRNA splicing and intronic polyadenylation in human tissues. To do that, I first identified polyadenylation sites based on poly-A reads found in GTEx dataset. Then I used coverage drop at polyadenylation sites to identify tissue-specific rate of usage of this pass. Then I used the same RNA sequencing dataset to identify the tissue-specific rate of alternative polyadenylation, which I could match with tissue-specific usage of the pass. This allowed me to predict alternative splice events associated with the intronic polyadenylation sites. And first, we used this dataset to characterize the kinetic interaction between pre-mRNA splicing and intronic polyadenylation. Accordingly, my talk will be divided in two major parts. First will be dedicated to the methodology of polyadenylation sites identification from RNA sequencing data, and the second will be dedicated to characterization of the identified polyadenylation site in GTEx tissues and simultaneous analysis of tissue-specific alternative splicing and intronic polyadenylation. We defined poly-A reads as reads that contain soft clip, which are at least 6 nucleotides long and contain at least 80% adenines. From each polyadenylation poly-A read, the position of first non-templated nucleotide can be used to identify the potential position of the polyadenylation site. Then these polyadenylation sites can be pulled between the samples, and at the end, each candidate polyadenylation site is characterized by first the number of corresponding poly-A reads, and second by distribution of lengths of the soft cleaveds, which we call overhangs. Our confidence in each polyadenylation site depends not only on the number of poly reads that support them, but also on the diversity of these reads, which can be measured as entropy of the overhang length distribution. Analysis of GTEx dataset yielded about 500,000 polyadenylation sites, and an example of distribution of poly-A reads around one of annotated transcript ends you can see on the figure. I want to note that the y-axis here is in logarithmic scale, meaning that this cluster of polyadenylation sites have two major paths, which have much higher coverage of poly-A reads than the other parts located around them. These clusters of polyadenylation sites were not uncommon. Most of the transcript ends were surrounded by a set of paths rather than one specific peak. This tells us that the cleavage and polyadenylation machinery may be imprecise and often introduce cleavages around the main polyadenylation site. Additionally, we try to estimate and look for determinants of width of the polyadenylation clusters. Interestingly, the only significant determinant factor that we found is the presence of the polyadenylation signal upstream of the site, meaning that if the polyadenylation signal is there, then normally the cluster width is smaller, meaning that the cleavage and polyadenylation is more precise. Since the polyadenylation sites clustered together, we merged adjacent paths and formed path clusters. So as a result of analysis of GTEx, we got 300,000 GTEx-based polyadenylation sites clusters. To validate this set, we first compared it to annotated transcript ends from gene code and to a polyadenylation site set from polyadenylation atlas that was previously generated and published from 3' end sequencing libraries. As you can see in the figure, the precision of our GTEx-based path set, which is yellow, is slightly higher than the precision of the polyadenylation sites atlas. However, the recall is slightly lower. This tells us that the set of polyadenylation sites inferred from GTEx dataset is comparable to the set of polyadenylation sites from hundreds of 3' end sequencing libraries, and good enough for further analysis. However, I want to remind that RNA sequencing data, in contrast to 3' end sequencing data, allows to simultaneously assess tissue-specific polyadenylation and alternative splicing, which is not possible for 3' end sequencing techniques. And this is what we're going to do in the second part. Specifically, first, I will characterize the identified polyadenylation sites, and then use tissue-specific alternative splicing and intronic polyadenylation rates to match them and to match the patterns of tissue-specific alternative splicing and intronic polyadenylation. About 170 polyadenylation sites, polyadenylation sites were in protein coding genes. Each protein coding gene can be divided into several parts. These are two untranslated regions, 5' and 3', open reading frame in the middle of them, and the open reading frame can be further divided into constitutive exons, introns, and alternative exons. Next, we examine the distribution of polyadenylation sites within these regions. First, as expected, the majority of polyadenylation sites fell within 3' untranslated regions, since these are the polyadenylation sites that are annotated and the polyadenylation sites that are in the same 3' untranslated regions. However, A lot of polyadenylation sites also are located in open reading frame regions, meaning that these are novel polyadenylation sites that are unannotated. On the other hand, since the open reading frame regions are much longer than 3' untranslated regions, the density of polyadenylation sites in open reading frames is much lower than in 3' untranslated regions, which is expected because most of cleavage and polyadenylation, the density of cleavage and polyadenylation sites in untranslated regions is higher. When we consider the polyadenylation sites that fall within open reading frames and subdivided them into ones falling into intronic, constitutive exonic, and alternative exonic regions, we surprisingly found out that the majority of polyadenylation sites from open reading frames fall within intronic regions. However, the intronic regions are also longer than exonic ones, thus the density of polyadenylation sites in intronic regions is lower than in exons. Still, we found more than 45,000 polyadenylation sites located in introns. It means that on average, there are 2.5 intronic polyadenylation sites per protein coding gene, which is a huge amount. And it again reminds us about the question of mechanics of silencing of these intronic polyadenylation sites. The polyarid-based approach allows us to also quantify the rate of cleavage and polyadenylation, since for each given polyadenylation site, the number of polyarids supporting the site would be correlates with rate of cleavage and polyadenylation events at this site. Thus, we can use polyarid count and its distribution to identify where the majority of cleavage and polyadenylation events happen. Again, not unexpectedly, the majority of polyadenylation events happen in 3' UTRs, and I want to note here that, as you can see here, almost all cleavage and polyadenylation events happen at polyadenylation sites which have polyadenylation signal. This happens because cleavage and polyadenylation rate at pass with signal is much higher than cleavage and polyadenylation rate at pass that do not have a polyadenylation signal upstream of them. And thus, pass with signal are supported by many more polyarids. Additionally, if we again look at intronic polyadenylation sites within open reading frames, the majority of cleavage and polyadenylation events are located within introns. However, their density is again lower in the intronic regions. The majority of introns are excised from the transcriptome by splicing, which means that if we want to compare a rate, cleavage and polyadenylation rate at intronic and exonic regions, we would somehow try to assess the density of polyarids that would have been observed in introns if they were not spliced out. To do that, we estimated the effective density of polyarids by normalizing number of polyarids by read coverage at the corresponding regions, and compared it with the effective density of polyarids within exonic regions. As you can see on the right, the effective density of polyarids within intronic regions is higher than that within exonic ones, which suggests that somehow rate of cleavage and polyadenylation at introns that do make it into the transcriptome is higher than in exonic regions. Another way to compare the rate of rate of cleavage and polyadenylation events within intron and rate of cleavage and polyadenylation events within exons accounting for the bias created by splicing is to compare the polyarid densities in intronic and exonic regions with similar coverage. To do that, we picked about 2,500 intronic and exonic regions with similar coverage and compared the density of polyarids within these regions. Again, not only polyarid count, but also polyarid density was higher within intronic regions than within exonic ones covered by the same number of reads. This shows us that the effective cleavage and polyadenylation density within intronic regions is somehow bigger than that in the exonic ones, which begs the question first, why is that? And this is one of the questions that we try to answer in the following research. So to answer this question and to go on with analysis of intronic splicing and alternative polyadenylation, I picked 36,000 high confidence intronic polyadenylation sites and analyzed the read coverage around the site and in the exonic region, which is upstream of each intronic polyadenylation site. Using this data, I selected more than 7,000 intronic polyadenylation sites that are used in the GTEx tissues. To do that, I first selected sites that have substantial read coverage upstream of the pass here and at least 10-fold coverage drop at the pass, meaning that the coverage changes substantially after the polyadenylation site. Each intronic polyadenylation event leads to an expression of alternative terminal exon. Specifically, there could be two outcomes of each intronic polyadenylation event. First is expression of retained intron, partial intron retention. In this case, an alternative terminal exon is referred to composite terminal exon. Otherwise, intronic polyadenylation event can lead to expression of a cassette exon. Thus, this type of alternative terminal exon is called skip terminal exon. And since any intronic polyadenylation event is associated with one of the two, the intronic polyadenylation events themselves are often also called composite and skipped, according to the type of terminal exon that they are accompanied by. One of the ways to identify the type of alternative terminal exon is examining the coverage in the beginning of the intron. Specifically, in case of skip terminal exon or expression of a cassette exon, the coverage in the beginning of the intron would be negligible compared to the exonic one. However, in case of composite terminal exon, which is partial intron retention, the coverage in the beginning of the intron would be comparable to the exonic. Here in this work, I refer to coverage in the beginning of the intron as WE2 and at the end of the exon as WE1. Part of the intronic polyadenylation sites that are annotated can be classified as skipped or composite solely based on the gene code annotation. Thus, we picked these events and compared the coverage in the beginning of the intron and in the end of the exon for these already classified events. As you can see in the figure, expectedly in case of skip terminal exon, the coverage in the beginning of the intron is lower than coverage than the exonic coverage. However, in case of composite terminal exon, the coverage in the beginning of the intron is very similar to the exonic ones. Based on this analysis, we picked a threshold of 0.25 ratio between WE2 and WE1 to distinguish between the types. Interestingly, the annotated set of intronic polyadenylation events also contain the same two groups, meaning the group with coverage where WE2 is almost equal to WE1, and the group with WE2 is less than WE1. Remembering the ratio coverage that can be used to clearly differentiate between the annotated types, we also can apply this ratio and identify the type of the terminal exon for these unannotated intronic polyadenylation sites. Now I move on to another more common way to study alternative splicing, which are split reads that align to splice junctions. Specifically, for any intronic polyadenylation site, I picked the shortest annotated event, the shortest annotated intron that contains this polyadenylation site. For this intron, I identified, I counted the number of split reads supporting exclusion of the shortest annotated intron, referred here as A. Then I counted number of split reads supporting the exclusion of a shorter intron that ends upstream of the polyadenylation site, and number of reads, of continuous reads that support intron retention. These numbers allowed me to quantify what I call rate of canonical splicing, or psi. The rate of canonical splicing shows fraction of the transcripts where this shortest annotated intron containing the pass is excised. This data compared with data of intronic polyadenylation rate, which can be identified from poly-A read support of the polyadenylation site or coverage drop at the polyadenylation site, can be used to look for correlation between intronic polyadenylation rate and rate of canonical splicing. On the left, you can see an example for one intronic polyadenylation site. And in the plot, we can clearly see that intronic polyadenylation rate measured by poly-A read support and canonical splicing are negatively correlated. This is also true for all intronic polyadenylation events that we identified in our dataset. Specifically, the higher the psi, the lower is poly-A read support, which means that rate of canonical splicing is negatively correlated with the rate of intronic polyadenylation, which is expected since if the canonical here intron is excised, then the intronic polyadenylation site is also excised and should be degraded as part of the intronic lariat. The same was true for annotated intronic polyadenylation events that could be classified as skipped or composite based on annotation. In other words, annotated intronic polyadenylation sites have low canonical splicing rate in tissues where they are expressed or used. However, when we looked at intronic polyadenylation sites that could not be classified as skipped or composite based on the annotation, we saw an additional peak at psi equal to 1. This peak means that these intronic polyadenylation events are not associated with any alternative splicing event, and thus this peak is incompatible with both composite and skipped models, since it means that these are a group of intronic polyadenylation events that are accompanied only by canonical splicing. Since this is pretty unexpected, we picked this group of intronic polyadenylation events, which, by the way, do not contain many non-annotated transcripts, meaning that they are not ends of known transcripts that end within introns of other transcripts. So we picked the group of these events and tried to study them further. Specifically, we analyzed the coverage in the beginning of the intron, of which I've talked in the beginning of the talk. Interestingly, we observed that for this group of events, coverage in the beginning of the intron is very low, similarly to skipped terminal exon events. Thus, these are intronic polyadenylation events that are accompanied only by canonical splicing and characterized by low coverage in the beginning of the intron. However, what is different for them from skipped terminal exons is that there are no alternative splicing events that could explain this coverage. Specifically, to make it clearer, I'll show you a couple of examples. First is example of skipped intronic polyadenylation event that happens in attractin gene. We clearly see that there is coverage upstream of the intronic polyadenylation event, which drops at the intronic polyadenylation site. However, this clearly is a tissue-specific alternative terminal exon, since there are junctions that support this alternative exon or cassette exon. And these junctions are observed only in the same two tissues where the coverage of the alternative terminal exon is observed. However, if we look at an example of intronic polyadenylation event accompanied only by canonical splicing, we see that again there is coverage upstream of the intronic polyadenylation event. And in this case, it is not tissue specific, so it is observed in most of the tissues. However, there are no junctions that would support inclusion of an alternative terminal exon. Moreover, there is no coverage in the beginning of the intron, which would show us that this is a case of intron retention. Thus, I concluded that this is somehow a group of new intronic polyadenylation events that are incompatible with both previously known models. So to describe how these intronic polyadenylation events could appear, so we think that these events could appear because when cleavage and polyadenylation complex gathers on the polyadenylation site after the spliceosome has already recognized the intron and started the splicing process. This way, the last step of splicing would remove the products of cleavage and polyadenylation as part of the lariat, meaning that the lariat removed would have an excision in the middle of it. We called these events spliced polyadenylated introns and think that they are a result of interplay between splicing and polyadenylation events. Specifically, again, they appear when cleavage and polyadenylation happens after the spliceosome has already started the splicing process. And thus, we think that this expands the model that is normally, that is commonly used, and introduces events where both intronic polyadenylation and splicing work on the same intron. If that was the case, then we'd be able to observe spliced polyadenylated introns in some other more specific data sets. To do that, we first hypothesized what would happen with these SPIs in the cell. And we think that it will be first linearized by the branching enzyme, as well as any other lariat. And then the results of this linearization would be two RNA molecules, one of which would have a poly-A tail and 5' monophosphate. And this molecule would correspond to the beginning of the intron, where this intronic polyadenylation site was located. Interestingly, there already exist methods to capture these specific types of RNAs. One of these methods is called CleavSeq, and it captures 5' ends of polyadenylated RNAs with 5' monophosphate. We reanalyzed the CleavSeq dataset and indeed observed that the coverage of 5' ends of CleavSeq reads is higher in the beginning of the introns where there is an intronic polyadenylation site compared to introns that do not have polyadenylation site within it. Additionally, what is even more interesting, that after classifications of intronic polyadenylation sites into SPIs, composite and skip terminal exon ones, we observed that CleavSeq coverage is higher for in introns where we found SPI polyadenylation sites. Meaning that first, the CleavSeq dataset supports the existence of SPIs, and next, it again shows us that SPIs are not transcripts initiated and terminated within the same intron, at least since these transcripts are clearly not kept, not transcripts. Finally, as we have now these three types of intronic polyadenylation events, we can classify the intronic polyadenylation sites that we observed in the GTX tissue into skipped, composite, and SPI ones. For that, we first used the coverage in the beginning of the intron compared to the exonic one, which I described in the beginning. And then as SPIs, we picked the polyadenylation sites that have psi close to 1. As a result of this classification, we saw that more than about 90% of annotated and classified based on annotation intronic polyadenylation sites had correctly predicted types. And which is more interesting is that we saw that compared to numbers of other events, SPIs indeed constitute a substantial fraction of intronic polyadenylation events, and thus are an interesting addition to the model of intronic polyadenylation. So to conclude, I first remind that we identified a set of polyadenylation sites inferred from GTEx RNA sequencing dataset, and saw that this set is comparable to the one from hundreds of intronic polyadenylation sequencing libraries, hundreds of 3' end sequencing libraries. Next, after proper normalization, we saw that the relative density of polyadenylation events in introns is larger than in exonic regions, which reflects intrinsically higher cleavage and polyadenylation rates within introns compared to exons. And finally, a substantial fraction of intronic polyadenylation events are accompanied only by canonical splicing events. And we think that these events, which we refer to as spliced polyadenylated introns, are byproducts of interaction between cleavage and polyadenylation and splicing happening within the same intron. Taken together, this data shows us that probably indeed products of some of cleavage and polyadenylation events happening within introns are excised by splicing, and thus splicing could be a natural mechanism of suppression of premature transcription termination, which, if we look at it in a broader sense, somehow does not correspond to idea that the main and the only function of splicing is proteome diversity, which is a commonly accepted one. We hypothesize here that splicing could excise the introns where defected polyadenylation sites appear, and thus, similarly to NMD, be a mechanism that counteracts premature transcription termination. And so I want to thank all previous and current members of my lab for the advice and help during this project, and of course, thank my supervisor, Professor Dmitry Pervushin, who guided me through this whole research. Thank you all for listening and for asking interesting questions about the research, and I will be happy to discuss it further now. Thank you.
Thank you. We can start question and answer session. We start with question from the jury, probably in the same order as I have presented. Do you remember from Professor Jean, please, if you have any questions to discuss?
Yes, your microphone.
My mic is off. Once again, Professor Jean, I don't see him. Oh, yeah, here. Please, questions.
His mic is off.
Thank you. Okay, please go, Professor, please go ahead. Your questions.
Mine.
Yep, fine. We do hear you.
OK，那个Mariia, overall, this paper, this paper is very well structured, and rich in data.
Okay, it seems there is some problem with internet. Okay, probably we'll move on to the next jury member, and meanwhile, check what's going on with the internet here. I'm sorry. Professor Gusev?
Yeah, thank you very much. Can you hear me?
Yes, we can hear.
Yes, okay, right. So, Mariia, thank you very much. I like this high-throughput work. You did it very well. So I have two questions. I represent, say, biological wing of a jury, probably, as Mikhail mentioned, so more in this kind of biology things. I have two questions. The first one, you saw this alternative polyadenylation sites events inside of the genes, right? Not in the end, in some time. So number is not very high compared to the like proper one. But how do you think, is there any biological implication in that? Because you are suffering from the high throughput, so from high amount of data, you say, okay, this number of low, but what is it? Is it transcriptional noise? Or is it kind of logical, some biological event connection happening? What do you think? Because these days, the high throughput sequencing data is just growing. So do we ignore this low number, say, kind of marginal cases, or this is some biology behind it? What do you think?
Yeah, okay. Thank you for the answer. I think if you could, Yelena, if you could show the slides. So since you've asked the same thing during
Yes,
the
exactly.
I'm trying to move the slides and I'm not able to do that yet. So I'll start with the answer. First, indeed, there were many polyadenylation sites within intergenic regions, which, so you don't have to read that all. So just the table here that you referred to says that there are about 190 intergenic polyadenylation sites in intergenic regions. I did not focus on them in my research, but previous analysis of data from plants, yeast, and mammals have also reported similar, like a bit lower, but also very big numbers of polyadenylation sites in the intergenic regions. These often are referred to either antisense transcripts if they're located on if they have an intersection gene on an opposite strand, or also often they are thought to represent 3' extensions of known genes. But the extensions that are very long, so it is unexpected to see them this long. On the other hand, of course, several of them were found, several of similar intronic pollination sites were found to be ends of novel transcripts. However, normally these are numbers like 10 out of 10,000, so not high indeed. And of course, they also could arise from the pervasive transcription, and it's possible that some of them are artifacts due to analysis issues like misalignment. For the specific polyadenylation sites that I found in this research, there were 100,000 intergenic PAS, and they formed 122,000 PAS clusters. Surprisingly, which I analyzed it after your question, but interestingly, most of them are seem to come from antisense transcripts, meaning that almost 80,000 of the polyadenylation sites have a gene intersect with a gene on an opposite strand. This gene can be either coding or non-coding, but still this is the majority of these polyadenylation sites. Interestingly, density of them rises towards the end of the gene. So this calls probably for further research and to understand why is that. But indeed, it is an interesting thing that I observed. Additional 35,000 of intergenic polyadenylation sites are far from, do not have an antisense gene, right? However, they tend to, their density is higher downstream of the gene, meaning that they many of them represent these already mentioned very long 3' extensions of annotated genes. This density is higher downstream of the gene for up to about 15 kilobase downstream of the genes, which interestingly corresponds to previously found extensions up to 20-15 kilobase, which were found by in other studies also done on mammalian species. Yeah, I hope this answers your question. But indeed, there is.
Thank you. Yeah, I don't think it's a answer, but it's very nice elaboration. So thank you very much. It's very much appreciated. So the following question is: you used retext, so I mean, say collection of transcriptomics from different tissues, right? So collect large number of reads, but I probably missed it, so I apologize in advance. But how about this tissue-specific dynamic of the polyadenylation and splicing events? So you have a lot of tissue, right? So is there anything linked? So I mean, number of events of polyadenylation with specific tissue or specific state or specific organs. So do you have anything to say about it? It's quite interesting.
Yes, I don't think you've missed it. It was briefly mentioned in the thesis itself, but I didn't talk about it a lot. So first, I just want to state that all events that I've referred to during the presentation, I don't know where to go, but so anyway, for example, here, right? So all the events that we analyzed are tissue specific. So the coverages and the numbers of split reads, etc. This was all counts for past tissue pairs, meaning that it was each event is basically an polyadenylation event in a tissue, right? As I understand, you're asking about some specific examples of tissue-specific events. Since mainly we use the drop in coverage density to quantify the usage of the polyadenylation sites in the tissues, I did not focus on that because there have been already several publications that just focus on this type of thing. Indeed, we observed some tissue-specific events. I think there is an example in the thesis itself. However, it was not the focus per se.
Okay, thank you. And the final, very small question. So, you know, as Mikhail mentioned, we are keen on some strange creatures, right? And one interesting feature of some of these guys that ATGC content in a genome can be drastically different. So, for example, let's say Mitch study has extremely high AT ratio, but I mean, literally will kind of extend number of AAA or TTT repeats in the genome. So how do you think, depending on the balance of GT content in the genome, do you expect it will affect globally on the dynamics of alternative polyadenylation sites, because it's naturally provide more, say, sites for polyadenylation or to miss a proper one and have kind of alternative by mistake polyadenylation site. So what do you think of it? Of course, you don't have data because it was not focused of your study, but how do you think? If it's extremely AT rich genome, do you expect a global shift of the logic of the polyadenylation genome?
Thank you for this really thought-provoking answer. I'm not sure I thought about it a lot, but I mean, since we saw and uh, it was previously observed many times that this polyadenylation signal, which is essentially built of AAA and T and AAA, is crucial for the cleavage and polyadenylation mechanism and for recognition of the polyadenylation site. If the machinery is the same in that species with this other more AT-rich genome, if I got your question correctly, so if the cleavage and pollination machinery is similar and if it recognizes the same signal, I think indeed cleavage and pollination rate would be probably higher. However, I imagine that there would be some compensation mechanism in this case. I hope I got your question correctly. If not, could you?
Yes, to a large extent. Thank you very much. I'm all done, all questions answered properly. And thank you for addressing my questions to the thesis by making separate explanatory slides. Thank you very much, sir.
Thank you.
Okay, and there is a formal question whether you are satisfied with the changes made in the thesis following your report.
Very much, and Mariia very nicely incorporated slides answering my questions. So I totally satisfied. Thank you.
But that's a formal question that we have to ask. Okay, thank you. Maybe we can try once again to reach Professor Jean. I don't. He's connected. Okay, so could you turn him on? Elena?
The diameter interaction between alternative.
Yeah, it's better now.
And polyadenylation mechanism. Are there specific or regulated elements that play an important role in this process?
Well, it seems still to be some problem with the connection because we've heard you, but it was very difficult to hear specific words. So maybe if you have questions, maybe you can type them and Mariia will answer. I mean, you may type your questions in chat and Mariia will answer them if it's possible. Yep.
Okay.
Okay, yeah, thank you.
Yeah, I do not understand that. Could someone read that, because it is kind of impossible to read?
Yes, I will help you with that because I can see it on my laptop. So number one, in the study, you mentioned the dynamic interaction between alternative splicing and polyadenylation mechanism. Are there specific factors or regulatory elements that play a key role in these processes?
Is it another or okay? So I'll start with that. We did not look for specific factors that could play a role in the interaction between the process. However, as I mentioned, we know that there are many factors that do regulate both intronic polyadenylation and alternative splicing, and I suppose that they are the ones that could be could be candidates for these factors that play roles in their interaction. So, however, I did not study that indeed, and this calls for further analysis. I hope I understood your question correctly. Thank you.
Okay, I sent an And next question
Okay.
to you.
Okay, the next question is, based on the tissue-specific patterns you observed, do you think that the interaction between splicing and polyadenylation could serve varying functions in different tissues or cell types? Example, neuron, immune cell.
Yeah, this is an interesting question. I think I'll need, so if possible, I'll need a slide. Slide would help me to answer that.
What slide is it? Maybe I can help opening
I think
it.
you can just open and I will scroll if I can.
Okay, try now.
Okay. So we did not address this specifically, but one way to look at that could be looking at number of intronic polyadenylation events, amounts of intronic polyadenylation events classified as spliced polyadenylated introns in different tissues. And so as you can see on the bar plot on the right, these are numbers of intronic polyadenylation events identified as SPIs skipped to composite in different tissues. And we do not see any tissues where amount of intronic polyadenylation events that are SPIs would be considerably higher or lower than in other tissues. So while this suggests that there is no tissue that uses this type of regulation more often than others, specific events, of course, could be tissue specific, and this also could be studied further. I hope this answers.
Thank you very much. Thank you for your answer. Thank you.
Sorry, Professor Jean, just to confirm, any more questions to follow from you at the moment?
No. I have finished it. Yeah.
Okay.
Thank you for your questions.
Okay, and again, Professor Jean, the formal question of whether you are satisfied with changes in the manuscript and with the responses to your questions.
Yes.
Okay, thank you. Our next external jury member is Dr. Ivan Kulakovsky, please.
Hello, can you hear me?
Yeah.
Oh, perfect. First of all, dear jury members, dear Professor Gelfand, dear Mariia, it is a pleasure to read the text. It was really a pleasure to read the comments and see the changes being really introduced in the original, I would say, draft. So the final version is really beautiful, both in scientific and well, presentation sense. So I fully support it, and I have no further questions regarding the content, and I think it should be accepted. So I'm kind of running ahead and just answering the formal stuff. I'm fully satisfied, and I'm not going to re-ask any questions that I have raised. But I have two well, things that I would like to hear an opinion, Maria's opinion. One is purely scientific and the other is more technical. I will start the one that's related to, well, the biological processes that go in the cell. What we know, well, I'm more a transcription-oriented person, and what we know regarding cell type specific transcription, there are multiple alternative transcription start sites, and there are, well, often clustered in the vicinity of 5' ends of the gene, and the first introns carry, well, tens of alternative sites. Sometimes they are very highly active in particular cells. And then my naive vision on splicing is that once we start transcription in an inappropriate place, the splicing and the processing goes also astray. And well, anything could happen. There might be exons kept or spliced in a very weird fashion. And then I would expect that intronic, at least part of the intronic polyadenylation sites, will be coming from the transcripts that didn't use a major canonical TSS, but instead they started somewhere in the middle of the gene. So it's not related to start and end of the transcription in a single intron that you ruled out with CleavSeq, but it's more about aberrant transcription, like it started in the middle and then just used some downstream polyadenylation site, which was selected randomly. So it's a general transcription noise and not something that needs special machinery to like cut parts of the introns and then will run polyadenylation on in a different manner. So what's your opinion on this? Could we explain even a minor fraction of weird intronic polyadenylation sites just by considering non-canonical but still very active alternative transcription sites?
Okay, thank you for your question and for your wholesome support of the work. First, I would just want to mention that indeed there is research showing that specific transcription start sites transcripts starting at specific start sites tend to have other polyadenyl, like tend to end in different places than transcripts starting at another start sites, which is like also interesting since these are two events that are quite far away from each other, sort of. And again, to answer your question about the explanation of these SPIs that we see, as I understood it, I'm not sure, like maybe the picture of the example would slightly help. I'll try to, I think that this should not be the case. Because so we are looking at here we're looking at an annotated intron. Like normally the canonical splicing, which I refer to as canonical splicing, is the annotated intron. However, when analyzing the psi and looking for alternative splicing events, such as intron retention or cassette exons, we did not only look at, specifically in case of cassette exons, we didn't only look at splice junctions, split reads starting in the beginning of the intron. However, we also looked at split reads that would start in some other places and land upstream of the intronic polyadenylation site, which would like kind of be one, I think, the case that you are referring to, right? And still, so we did both, looking only on this type that start the junctions that start at the 5' splice site, which is annotated, and considering also the junctions that start upstream, and it didn't significantly alter the analysis. So maybe some of the events are indeed explained by these not known alternative starts, splicing events that start somewhere far away upstream and are referred to come from unknown transcripts that start in other transcript start sites. However, still, even if we exclude these, still there are events that have for which we do not see any ways that tie them to transcripts outside of the intron, sort of, if you understand what I mean. So I hope it at least partially answers the question. If not, could you elaborate, please?
Yeah. Thank you so much for the clarification and for proper illustration. Yeah, thank you so much. I have another question, more technical one. Well, most of us work with transcriptomics data. We usually start by grabbing some well and widely used genome annotation like gene code, which Professor Pervushin has contributed to a lot, and it's still imperfect. And once we start working with a particular tissue sample or cell type, there are many transcripts which do not align well with the fixed limited annotation, including, of course, transcription end sites and polyadenylation sites as well. So my question is, it's related both to alternative splicing and polyadenylation. Would it be possible to generate a robust atlas of sites that you detected by GTEx-wide analysis and maybe complemented with some user data? Like if I bring some custom transcriptomic data, short read or long read sequencing, then somehow merge it with your database or catalog of alternative pesticides and introns. And then could I get, let's say, GTF, a robust, simplified, of course, truncated, but still a better annotation of the gene models suitable for, well, my particular research project. So can we turn the stuff that you learned, a comprehensive and I would say quite complex picture of alternative polyadenylation into something technically useful and something simple to use for general people who don't care about all the minor variants, but still want something better than a single, gene code. Is it possible? And what's even more important, does it make sense or just maybe it's completely crazy idea?
Okay, thanks for your question again. If we start with, I think that important contribution to the transcript annotation would be these types of alternative terminal exons, right? I mean, these are the things that do appear in the transcriptome and have some functions, while these lariats, we suppose that they are just spliced out, and the function of this process is the fact that products of cleavage and polyadenylation are removed and do not affect the transcriptome. So, I mean, I'm not sure if this is important to include in some sort of user-friendly database. If we talk about the alternative terminal exons and of two common types, skipped and composite ones, I think first there surely were already attempts to link intronic polyadenylation events from, for example, the polyadenylation site atlas that I've mentioned, to different types of alternative terminal exons. And there are first works that do it, however, in a different way that I did. And I think there even is a paper where this identification was turned into some sort of database of alternative terminal exons. So if we talk about the common two types, this, partially this has already been done. And there are specific methods to do that based if we have a set of polyadenylation sites. And if we talk about the lariats, about SPIs, I'm not sure this makes sense a lot. So I hope this answers.
Yeah, thank you. Just one final small question. Of course, this has already been done with different approaches, but do you plan to do it with your atlas? Do you plan to, not just forget it about, put it in Zenode, I think, all the data, and that's a nice point about your work. All the data in the code is in public repositories, and I'm highly supportive in this matter. That's really good. But, do you plan to make a next step to make something easily usable for the community, or just you're okay and let other people do this type of job?
To be honest, for me, the more interesting part was this identification of the different type of intronic polyadenylation events. Thus, I didn't plan to make specific database of these terminal exons. And I think additionally, this would call for some benchmark analysis of different approaches to identify the type of the exon, which was not the focus here. However, the benchmark analysis would take quite a lot of time, and then I'm not sure now if I'm gonna do that.
Okay, once again, thank you so much. And again, I don't have any further questions or comments regarding the original kind of issues that I have found and asked in my review. So I fully support your work, and I think it's a very high standard, both scientifically and in terms of presentation. Thank you so much.
Okay, thank you. So that responded to this formal question already, so we can move on. And Professor Vankov.
Okay, thank you very much. And actually, I don't have any comments or questions. The work is just beautiful, and I had a couple of cosmetic change, kind of comments to the thesis, and they were satisfied. And so my answer to the formal question I'm satisfied with the changes in the thesis. Thank you.
Thank you for your comments and help during the project.
Okay, thank you. And Professor Khamilova?
First of all, thank you, Mariia, for the opportunity to read this beautiful thesis. I think that the quality of the text of the thesis is very high, and the quality of the research presented in the thesis is also very high from the methodological or from technical point of view. So when I wrote my report, I didn't have any questions regarding the methods and results presented in the thesis, because I think they're next to perfect by their quality. But while listening to your presentation today, I've got a couple of additional questions, or rather things to discuss. And they are in the direction of questions already asked by Professor Gusev and Professor Jean. So I'm also curious about the functional role of this intronic polyadenylation events. And the question is, or rather the thing that I would like to discuss, is the following. So when we think about the alternative splicing, just alternative splicing, we know that different tissues have very variable number of events of alternative splicing. For example, the human brain is known to have the largest number of alternative splicing events, right? And we think that this fact, it is expected, because human brain has complex functions, and to address this multitude of different processes that happen in the human brain, probably it needs to have a high number of different isoforms that can perform these different functions, right? So if this alternative polyadenylation events were functional, I would expect tissues, complex tissues such as human brain, to have increased number of alternative polyadenylation events. But according to your slide that you were showing in the answer to Professor Jean's question, we don't see any difference, any significant difference in the number of alternative polyadenylation events. So does it provide enough evidence to say that these events are not functional? What do you think about that?
So if you're referring to function of the SPIs that were generated like this, I don't know what to say, these RNAs that line here, I don't think that the SPIs themselves have some functions, as I think I mentioned somewhere. So I think they are interesting in terms that they are products of this interaction that we observe, and this way we can see that these two processes can like meet and interact with each other. However, the function of these specific RNAs, I don't think there is one, and partially because the numbers of them are not tissue specific, and yeah, so as you mentioned. Yeah.
So basically, these truncated transcripts that end at these alternative polyadenylation sites, they are rather noisy, and you did not find any examples of the isoforms stopping at this alternative polyadenylation, terminating at alternative polyadenylation site, that would represent some major fraction of transcripts of a particular gene, right? Or are there any examples where this truncated transcript ending at the alternative polyadenylation site would be at least a half of the transcripts of a gene, or constitute some substantial proportion at least?
Okay, first, I think if we're talking about the proportion, we could, like, I didn't look for that specifically, I guess, but we could estimate it from the coverage of in the middle of the intron, exactly, compare it to the coverage of the exon. For example, in this case, we see that they are somehow comparable. So the coverage here is for some reason quite high. However, so as to the other question, I think it is related, but I'm not very sure. Since we identified the type tissue specifically, it means that there could be intronic polyadenylation events that were classified, for example, as you can here's the Venn diagram for the intronic polyadenylation sites. And if an intronic polyadenylation site was classified as skipped in one tissue and as SPI in another, it would end up here in the intersection. So there are SPIs that in some tissues are used, right? So there are ends of truncated transcripts, as you said. So I see it as the fact that in some tissues, this is a functional pass and is used. However, in some other tissues, for some reason, splicing happens faster than in the other tissues. And then maybe, and then the cleavage and polyadenylation events happens. However, its products are spliced out, and thus it is an SPI and not a skipped terminal exon, for example. So this, as you can see, there are not many of such events, but still they're there, and maybe it calls for first, I don't know, most specific and I don't know, most scrupulous typization first, and then if we indeed find that there are tissue-specific SPIs, it would be interesting to look for functions and mechanisms that make them tissue-specific.
And another question is, you probably did not look at the cancer data, but if you did look at TCGA data, for example, in addition to GTX data, what is your expectation? Would you see more events there? Because cancer data, they usually have more noisy products, or this mechanism controlling the number of these unwanted transcripts. They maybe do not work very well in cancers.
Yeah, indeed. As you probably know, I was like, in the beginning, the plan was to also look at the cancer data. However, when we found this SPI, I think I kind of got buried in that. So I started looking at the TCGA dataset that you mentioned, and but I just used poly-A reads to identify the polyadenylation sites there, but I did not reach the typization and the tissue specificity and cancer type specificity of various types of intronic polyadenylation events. So what I can just say is that there are more, like, we see a lot of new polyadenylation sites in the cancerous data that are not present in the GTEx. I think if we merge, there's almost so I think there is about 900 plus clusters if we merge the data sets, and these two data sets, I think, intersect by something like 100,000 or so. So the intersection is there and is big. However, cancerous data adds a lot of novel polyadenylation sites that can be further analyzed in this sense too.
I wonder if there could be some correlation between particular cancer severity and the number of these events, because some mechanisms controlling these unwanted transcripts could be misregulated in these particular cancers.
Yeah, it could be true. However, unfortunately, I did not
have time to dive into that. It would be a nice extension of your work. And maybe the last question, did you just out of curiosity, did you look at any motives of any factors that could bind near these events, intronic alternative polyadenylation events that could potentially somehow relate?
I think at one point I analyzed YACLIP datasets and tried to, however, I think at that point I tried to look for transcript for RVPs which binding sites are enriched around just intronic polyadenylation sites, and I don't think I did it for these specific three types. And the ones that I enrich around intronic are, so of course, there are if we compare it with other introns, we see that there are many cleavage and polyadenylation machinery, which is in this intron, and there are no such binding sites in other introns, which is kind of obvious. So I didn't, include it anywhere, and at that time didn't go further in that analysis. However, analysis of motifs of factors or binding sites of factors that bind near different types could be an interesting thing to do too. Of course, well, even if we could get the tissue-specific data, it would be even better. However, there are no such thing as I know, but maybe I don't know everything in this area. So, yeah.
Okay.
Thank you for the question.
Thank you. I have no other questions.
Yeah, formal question better, yes.
Yes, I'm fully satisfied with this work.
Okay, and now it's my turn. I had a number of minor editorial remarks. They have been taken care of. There are a couple of things that might be interesting to discuss, more like a scientific discussion rather than questions about this specific work. So at some point you mentioned that non-consensus variants of the polyadenylation signal, polyadenylation motif, which is AAUAAA. So you mentioned that non-consensus, that the deviations from the consensus might be required for fine-tuning the level of polyadenylation at this particular site. If so, one might expect that these non-consensus variants should be conserved when you compare like different primates or even different mammals. Did you think about that at some point? Because we've seen that in splicing, actually. We've seen that non-consensus variants of splice motifs tend to be conserved.
Yeah, so again, this is the question that you mentioned in the review. And so as I mentioned in the answer, I think this is the right one. So this prompted me to look for some research that would somehow related to that. First, the hypothesis that the non-consensus signals were selected in evolution to ensure specific rate of cleavage and polyadenylation came from a couple of papers that I read, and there it was based on the fact that frequency of this frequency of these events and of these sequences, sorry, in the genome corresponds to their effectiveness in terms of cleavage and polyadenylation, which is interesting and maybe does call for this conclusion. However, it was only a hypothesis, and I think analysis presented in this paper from 2016 kind of tried to answer it, but this is the correct slide. Yes. So the authors they didn't analyze the data between 4 different species. However, they analyzed allele frequencies in human population. And what they did was they first took the ancestral human genome and then identified polyadenylation sites there, and including the non-consensus ones, meaning that they didn't identify only AAT, AAA, but about a dozen of other sequences which are known to be polyadenylation signals. However, they are much less effective.
Sorry, but that's a slightly different point from the one I am making, because what I'm saying is that one would naively expect that deviations from consensus are bad, and there should be positive selection towards consensus. So the question is whether we observe an opposite situation, whether we observe stabilizing selection on non-consensus nucleotides.
Yeah, I think, so I was kind of getting to that. So the thing is that, yeah, moving on, they studied the SNPs within the polyadenylation signals, both consensus and non-consensus ones, and they, yes, as stated here, they looked at 3 classes of SNPs: disrupting, which from which the polyadenylation signal dies, sort of. So it was a polyadenylation signal, and it is no more a polyadenylation signal. Impairing, which reduces the effectiveness of the polyadenylation signal, and improving, which for, like, I think for your question, are the most interesting here, right? So these are the SNPs that increase the effectiveness of the of this polyadenylation signal. And so the question, it would be interesting to see if we have negative selection against the improving mutations, right? I think it at least partially addresses the question that you're interested in. And surprisingly, why not? The authors did not observe any evidence. So they observed evidence of negative selection against disrupting and impairing SNPs. However, they did not observe any evidence against the improving SNPs. So there are, at least this data suggests that there is no such
but these are SNPs, and what about divergence?
Yeah, divergence is a different thing, and they did not analyze it. They just looked at fast cons rates.
I see divergence in the title of the paper.
Yes, so I'm telling you that divergence in the title refers to fast cons data. You know, it's a common track. And they also analyzed it. Again, in there, they did not see any evidence for negative selection. However, I find that fast cons is, it is not very nucleotide specific, you know, it is quite wide. The peaks there are quite wide. So if we're looking at SNPs at specific positions, I don't think it was very useful there. And indeed, they still did not find any negative selection.
Okay, but then this argues against your
it kind of
hypothesis.
That hypothesis.
Sort of, yeah. Okay. Just a question. I'm curious. You mentioned that 3' sequencing tends to miss alternative polyadenylation signals in highly expressed genes. Why?
Again, I think you referred to this figure, which talks about comparison of 3' sequencing and RNA sequencing data set, which I used for benchmarking.
And the difference is sort of concentrated in highly expressed genes. So I'm just curious what's possible explanation.
So you stated in the question, you say that I said that it tends to miss
I would
genes.
naively expect that the higher is expression, the more the data you have, and the more reliable would be any procedure, experimental or computational or whatever.
Yeah, indeed, it does make sense. And so I did write in the thesis that it tends to miss. And after your question, I understood that the word tends here is kind of too strong. So we just see that the 3' sequencing analysis misses some of the highly expressed paths, which are paths in highly expressed genes, which are here. So these are the ones found only by RNA sequencing data set. However, as I thought further about that, I think it's just, and as I stated in the answer, I think it is, in this case, it's just an artifact of the analysis, since the data that I got, 3' sequencing data that I got, so the poly-A's were already trimmed there, so it was hard to determine the strand from where it came. Thus, I used the genes with which this very path intersects to identify the strands, which made me exclude several thousand genes. And I think these are these very
I see. So that's a problem with the procedure rather than the nature.
Thing here, yeah.
Okay, thank you. And then again, you mentioned that alternative polyadenylation in intergenic regions. I mean, the signal coming from alternative polyadenylation in intergenic regions is the result of pervasive transcription just, transcriptional noise that gets polyadenylated. So the question is whether these noisy transcripts are spliced as well. If they are polyadenylated, one would expect that they might be spliced also.
For that, I truly have no answer. So first, as we discussed in the beginning with Professor Gusev, there could be not only results of pervasive transcription, but also antisense transcription or extensions of 3' unknown genes. However, in terms of this noisy pervasive transcription pass, which I think would constitute about 20,000 pass, as I remember correctly, which are far from all known genes. I don't have any, so if they are indeed
no, it's just, it's stuff for discussion.
Be...
I understand that you didn't look at that specifically, but it still might be interesting to see to what extent this noise is processed further. Noise is beautiful. We've discussed that in the course.
I remember in the course,
a lot. Okay, and the final question, again, it's not a question, it's rather a matter for speculation. Would you, if you had time and whatever, would you like to repeat this analysis using long nanoporids or something like that?
Yeah, since...
Do you expect that the use of long reads might improve some analysis or change something?
To be honest, here in terms of identification of the types of alternative terminal exons which are transcribed, so the ones that are not SPIs, it could be since this reads the long read sequencing data is often used to identify specific transcripts, which of course makes sense. This would be an extension. However, since this classification of known types is not per se the main focus of the research, I don't think I would redo it with the long reads, since in case of SPIs, the long reads would not help us. Otherwise, I'd be if I pick the data that I could use some other data, I would, for example, more look for, there are specific ways to do Lariat sequencing, so it would be interesting to do Lariat sequencing with poly-ANs, like poly-A plus Lariat sequencing, which was not done because no one expects Lariat to have poly-ANs. And this, for me, would be a more interesting thing because I could see the SPIs. However, long reads in this particular sense, I don't think it would help in any way.
Okay, thank you. Then that's it from my part, and I am also satisfied. Any questions from the audience? 1, 2. I see none. Then Professor Pervushin, please.
Thank you very much. I think I am supposed to evaluate both work and the defendant, and I will start with a few words about this work that basically started as a blue sky project of what can we squeeze out of GTX data in terms of these poly-A reads and where in principle it can get us. And this work was not born very easily, and it was a difficult one, and it's a really complex one. I think it contributes significantly to the understanding of landscape of polyadenylation. And Masha did everything very well, and she is a fantastic PhD student. She is completely independent, and many of the, so for example, I didn't control some parts of what, I truly worked as a supervisor directing her towards goals, which she achieved herself. She is a completely grown-up scientist, I think ready for the future career. I must note that she is very thorough and very fundamental, and everything she does, she understands. And if she doesn't understand something, she refuses to do. And you can also see it from the way she answered the questions, that she is completely grown up, and it was a great pleasure to have you. with me 4 years. So I think she fully qualifies for a PhD title, and congratulate her with this fantastic and interesting work. I think this work will bring you more surprises in the future by citations. I mean, it will be highly cited, I think.
Okay, then we go to a breakout room in Zoom, and we probably have to ask the students and Mariia and the audience to leave us alone for a while. We'll invite you when we are done with our deliberations.
For your questions and comments.
It's not time for that.
Audience online, we're ready to start the closed deliberation. I would ask everyone to stay connected. I will now only open a separate breakout room for the jury members. Yes, Professor Perushin also needs to leave. So, dear jury members, just please make sure to accept the invitation when you see it in several, maybe in another 30 seconds.
Okay. Dr. Vlasenok, we congratulate you on your beautiful defense, and Professor Frischman, do your job. You have to pass the cap and the gown.
I just want to thank first, of course, my supervisor, who very thoroughly guided me and supported me through all this research and all this 4 years, which sometimes were not very bright. However, yeah, thank you very much. And again, thank you for all the reviewers for your comments. Some of them were really thought-inspiring and provided me with ideas for future perspectives of the research. So thank you for your thoughtful reading and support. That's all.